Good morning, guys. All praise to the Most High. Coming to you guys with a quick message. Found this very interesting. Popped up on my little news app. This is from yesterday, guys. And it says, scientist says Earth atmosphere is raining viruses and bacteria down on the entire planet. If you'd like to start out your day by opening your window for a big breath of fresh air, you might want to rethink your habits a bit. A new study reveals that viruses are raining down from above on a non-stop basis, no matter where you live on the planet. As it turns out, Earth is kind of a gross place. The research was published in the International Society for Microbiology journal, found that Earth's atmosphere is really good at transporting viruses all over the planet. Hundreds of millions of viruses are pushed skyward by winds, carried aloof for thousands of miles before tumbling back down on whatever and whoever happens to be hanging out below. Every day, more than 800 million viruses are deposited per square meter above the planetary boundary layer. That's 25 viruses for each person in Canada. University of British Columbia virologist Curtis Suttle explains roughly 20 years ago, we began finding genetically similar viruses occurring in very different environments around the globe. This preponderance of long resistance viruses traveling the atmosphere likely explains why it's quite conceivable to have a virus swept up into the atmosphere on the continent and deposited on another. The researchers gathered samples from a high altitude by traveling to the Sierra Nevada mountain range in California. There they found billions of viruses and bacteria that were riding high, many of which are thought to have been pushed skyward by powerful ocean sprays. The viruses and bacteria then fall back to Earth, often thanks to weather events like rain or even dust clouds. The biological bits are often carried in the troposphere, which the researchers know is below the stratosphere, where commercial jetliners cruise but higher than the clouds. Obviously, the viruses floating high above our heads and eventually landing all around us aren't all bad. The vast majority are incapable of attacking human cells anyway, and most viruses that are simply come and go before we even notice them. As for bacteria, our bodies are havens for all kinds of bacteria that do us no harm whatsoever. Scientists have estimated that the average human body is home to as many as 1 trillion bacterial cells, many which we couldn't live without. Now, they're trying to play it off as that, guys, but this flu is not the flu. This flu is not the flu. They did something. They sent the bacteria. Why NASA is sending bacteria into the sky on balloons during the eclipse. As the moon blocks the sun's light completely next week in a total solar eclipse, more than 50 high-altitude balloons in over 20 locations across the U.S. will soar up to 100,000 feet in the sky. On board will be Raspberry Pi cameras, weather sensors, and modern Raspberry Pi cameras, weather sensors, and modern modems to stream live eclipse footage. They also have metal tags coated with very hardy bacteria because NASA wants to know whether they will survive on Mars. Every time we send a rover on the red planet, our own microorganisms latch onto them and hitch a ride across space. What happened to these bacteria once they're on Mars? Do they mutate? Do they die? Or can they continue living undisturbed, colonizing worlds other than our own? To answer these questions, we need to run experiments here on Earth, and the eclipse on August 21st provides the perfect opportunity. The balloons are being sent up by teams of high school and college students from across the U.S. as a part of the Eclipse Ballooning Project led by Angela Desjardins of Montana State University, where when Jim Green, the director of planetary science at NASA, first heard that over 50 balloons were being flown to the stratosphere to live stream the eclipse, he couldn't believe his ears. I said, "Oh my God, That's like being on Mars. NASA couldn't pass on the opportunity. The upper parts of the Earth's stratosphere, just above the ozone layer, is very much like the surface of Mars. It's about minus 35 degrees Fahrenheit with very rarefied air and is hammered by the sun's ultraviolet radiation. During the eclipse, conditions will get even more Mars like. The temperatures will go down even further, and the moon will buffer some of those ultraviolet rays to better resemble the radiation on the red planet. It's really quite an outstanding astrobiology and planetary protection experiment, Green says. The bacteria that will fly to the edge of space is a particular strand called, I don't know how to pronounce this, but you know what we're going to copy that so we can look that up. It was first isolated from soil outside a spacecraft assembly facility at the Kennedy Space Center in Florida in 1973. An astrobiologist at NASA's Jet Propulsion Laboratory. These bacteria form shields of spores that allow them to survive even when conditions turn deadly. It takes around 140 hours at 257 degrees Fahrenheit to kill 90% of these bacteria. These are some of the most resistant types of bacteria that we know of. Last week, why would you send the most resistant type of bacteria that you know into the air, knowing that people down here is going to get sick? What is wrong with these folks? Okay, I guess. Last week, Smith finished mailing the bacteria, which are not dangerous for people or the environment, right, to the student groups. But it's not dangerous, but you said it's some of the most resilient types of bacteria, so that means that hard to get rid of, which are not dangerous for the people or the environment. Okay. Only 34 of the balloons will carry the bacteria. The microorganisms are dry onto the surface of two metal cards the size of the dog tag. One card will fly to the stratosphere while one will remain on the ground to function as a control group. But can you get her a bottle? The balloons will launch every 15 minutes or so from states that are in the path of the moon's shadow. They'll fly for about 2 hours, reaching the stratosphere and eventually popping because of the pressure drop. Once they're back on the ground, a parachute will slow down descent. The students will track them by GPS, recover the metal tags, and mail them back to NASA. That's when, however you pronounce this name, and Smith will get to analyze how many bacteria have died and whether their DNA has changed in any way. Well, is they changing our DNA? If some of them survive the flight, that might mean that these bacteria may have already survived a trip to the red planet as hitchhikers on Mars on a Mars rover. We don't know for sure. It's actually this word. It's actually on any Mars rover. It was found outside the spacecraft assembly facility, not on the spacecraft themselves. But even if it's not, learning more about these resilient bacteria can help us understand how similar ones could behave on Mars and help NASA better understand the risk of infecting other worlds. Okay, so even if this wasn't found on on Mars, the essay, but even if it's not, learning more about these resilient bacteria can help us understand how similar ones could behave on Mars. If it's not from Mars, it can't teach you nothing about Mars. All you doing is killing us with these bacteria y'all finding and putting them everywhere. It helped NASA better understand the risks of affecting other worlds. After all, we send $1 million spacecraft to other planets and moons to search for alien life, so it makes sense that we want to make sure these places are protected from Earth's germs. Y'all want to protect them from Earth germs and not us from their germs? That's a little bit backwards. Pushing organisms to known limits of life can also help NASA find that life can also find that life. If we know that resilient bacteria can withstand certain conditions, then we won't look for life when those same conditions are found on other planets, Smith says. Right. NASA has conducted very few experiments with high altitude balloons and none with this particular strain of bacteria. So, flying over 30 balloons at once under such perfect Mars like conditions that won't be possible to replicate in the lab is an amazing opportunity. I don't think it's ever been done in terms of coordinated astrobiology experiment happening across the entire continental United States on the same day. Smith says this is a spatial coverage that one could never dream of in any other circumstances. Green hopes the experiment will also inspire the next generation of astrobiologists and planetary protection officers. He got into science when he was in high school and had the chance to use an observatory telescope to observe the sun. Taking part in the eclipse ballooning project might do the same for students flying the balloons. You never know what turns kids on, Green says. Okay, but that was something weird to say, but okay. What is this? What is this, guys? Forming bacteria originally included within the genus Bacillus and then reclassified as a separate genus in 1993. Bacteria belonging to this genus have been detected in a variety of environments such as soil, water, vegetable matter, rhizosphere, I'm sorry, I skipped that word, forage, and insect larvae, as well as clinical samples. The name reflects Latin "pene" means almost, so the pene bacilli are literally almost bacilli. The genus includes P. larva, which is known to cause American foul brood in honeybees, which is capable of fixing nitrogen. So it is used in agricultural and horticultural. Okay, what is this? JDR2, which is known to be a rich source of chemical agents for biotechnology applications and pattern forming strains such as P. Fortex and P. Now, y'all know how I am with these words. then, so I ain't even gonna mess with it. Discovered in the early 90s, which are known to develop complex colonies with intricate architectures, as shown in the pictures. Okay. So this is something that I will have to do research on, read a little bit up on it, and really figure out what it is and come back to you guys with it. But the fact that it said biotechnology, it say, has been rapidly growing since many were shown to be important for agricultural and horticultural, industrial, and medical applications. These bacteria produce various extracellular enzymes ens- such as polysaccharide degrading enzymes and proteases, which can catalyze a wide variety of synthetic reactions in fields ranging from cosmetics to biofuel production. Various also produce antimicrobial substances that affect a wide spectrum of microorganisms such as fungi, soil bacteria, plant pathogen, pathogenic bacteria, and even important anaerobic pathogens such as Clostridium. More specifically, several species serve as efficient plant growth promoting PGPR, which completely colonize plant roots and can simultaneously act as biofertilizers and as antagonists. Antagonists, not protagonists. Antagonists is bad, y'all. Of recognized root pathogens such as bacteria, fungi, and nematodes. They enhance plant growth by several direct and indirect mechanisms. Direct mechanisms include phosphates, subutilizations, nitrogen fixation, degradation, degradation of environmental pollutants, and hormone production. Indirect mechanisms include controlling phytopathogens by competing for resources such as iron, amino acids, and sugars, as well as producing antibiotics or lytic enzymes. Competition for iron also serves as a strong selective force determining the microbial population in the rhizosphere. Several studies show that PGPR exert their plant growth promoting activity by depriving native microflora of iron. Although iron is abundant in nature, the extremely low solubility means that most organisms face the problem of obtaining enough iron from their environments. To fulfill the requirements for iron, bacteria have developed several strategies, including the reduction of ferric to furous ions, the secretion of high affinity iron chelating compounds called pyrophores, and the uptake of heterologous. Y'all, I hate these medical words, and I done been to school for I'm a certified CNA, and I'm going to school to be an LPN, and I hate these words. I hate pronouncing them unless you gotta hear somebody who know how to pronounce them say it first, and then you be all right. But yeah, candidas causes. Okay, what is What is this? So, yeah, guys, it's not the flu. It's not the flu. I'm gonna have to do some more research on all of this because I was coming just to bring y'all those two news clips, and then I got into what is this bacteria that they sent up. So I'm gonna do some research on this bacteria, and then I'll come back to you guys with more understanding. Yeah, I'm gonna do some research on this, and I'll come back with more understanding. But guys, the main point of the video is it's not the flu. It's not the flu. My daughter had it. I took her to the hospital. They tested her for the flu. The flu came back negative, but they said that it's possible that it come back negative because it's so many different types of the flu or strands. I just believe that it's not the flu, guys.